Good evening, guys. Welcome back to my channel. Today, I'm going to talk about DNA, its structure, application, and repair. What is a DNA? DNA, deoxyribonucleic acid, is the hereditary material in all living organisms. It stores genetic information which is essential for growth, reproduction, and cellular function. It was discovered by James Watson and Francis Crick in 1953. It has a double helix model. So, guys, what is a DNA? DNA actually stands for deoxyribonucleic acid. The full form of DNA is deoxyribonucleic acid. It is the molecule that carries the genetic instruction of all living organisms. Its structure, it's a double helix, like a twisted ladder. Its structure is made up of, basically DNA is made up of sugar phosphate backbone and nitrogenous bases. The nitrogen bases are adenine, thymine, cytosine, and guanine. Base pairing is A pairs with T and C pairs with G. What is the function of DNA? Well, DNA stores instructions for making proteins which perform most functions in cells. The structure of DNA. As I said, it is a double helix made up of two antipolar strands composed of nucleotides, sugar, phosphate, nitrogen bases, and base. Then it has bases adenine, thymine, cytosine, guanine, and it has a base pairing of A2 2H bonds and CG 3H bonds. Sugar phosphate backbone provides stability. I will also show you the structure of DNA. Guys, this is the structure of DNA. You can see these are the sugar phosphate backbone. You can see CG. This is the base pair over here, and then these are the nitrogen bases. You can see guanine, cytosine, thymine, and adenine. Nitrogen bases basically are the building blocks of DNA that form the rungs of the DNA ladder. They're responsible for storing genetic information through their specific pairing. There are four types of nitrogen bases, and each of them are really important to know. First one is adenine, then there is thymine, then there is cytosine, then there is guanine. Also, you can say guanine, cytosine, thymine, and adenine. Base pairing, there is a base pairing rule, which is complementary base pairing. Like A pairs with T, which is 2 hydrogen bonds, and C pairs with G, which is 3 hydrogen bonds. This pairing is specific and helps keep the DNA double helix stable and consistent. So it is these pairs are very much important to keep the DNA double helix stable and consistent. Types of DNA. There are types of DNA. First one is A DNA, which is right-handed dehydrated form. Then there is B DNA, which is in most common form right-handed helix. Then there is Z DNA, left-handed, plays a role in gene regulation. Actually, DNA, if you want to really understand DNA, think of DNA like a zipper. The side of the zipper are the sugar phosphate backbone. The teeth of the zipper are the base pairs AT and CG, and the base pairs zip together through hydrogen bonds to hold the DNA together. So the third type of DNA is ZDNA, left-handed, plays a role in gene regulation. DNA replication. Now guys, what is DNA replication? Process of copying DNA before cell division, semi-conservative. Each new DNA molecule has one strand, and one new strand occurs during S phase of the cell cycle. So basically, DNA replication is the process by which DNA makes an exact copy of itself. It happens during cell division. There are various steps which happen during the DNA replication. The first one is the unwinding, which is the double helix is unzipped by an enzyme called helicase. Then the second step is the base pairing, where new complementary bases are added by DNA polymerase. Then the third step is two identical DNA molecules are formed. It's semi-conservative. Each DNA has old strand and one new strand. So this is what DNA replication is, and these are the steps of DNA. The first step is unwinding, the second step is base pairing, the third step is two identical DNAs formed. And then there is steps in DNA replication. I said the first one is initiation, where I said that an enzyme called helicase unzips the double helix. So DNA helicase unwinds double helix at the origin of replication, elongation, DNA polymerase synthesize new strands 5 to 3, then leading strand, continuous synthesis, leading strand, Okazaki fragments joined by DNA ligands, and the termination replication ends with entire DNA is copied. Enzymes in DNA replication. Helicase. What are the enzymes that is used in DNA replication, guys? This enzymes are really important to memorize well, because it has a huge importance in DNA replication, and it has a huge hand in DNA replication. These enzymes are like really important, and we all must know the names of this enzyme which takes place while replicating DNA. So helicase unwinds DNA strands, primase synthesis, RNA primers, DNA polymerase, add nucleotides, ligase, joins, Okazi fragments, topoisomerase relieves supercloning, then single-stranded binding protein stabilize unwound strains. DNA repair mechanism. Direct repair corrects damaged bases correctly. Excision repair, damaged base nucleotides are removed and replaced. Mismatch repair, fix errors after replication. Recombinational repair, use homologous DNA sequence to repair breaks. SOS response, images system in the bacteria. So basically, guys, what is DNA repair? What is DNA getting repaired from? So DNA repair is a set of process by which cell fix damage to DNA. DNA can be damaged by, now as I said, DNA can be damaged by UV lights, chemicals, and radiation. So the DNA repair process is a set of process by which cell fix damage to DNA. Also, it gets destroyed by radiation and replication errors. How does a DNA get repaired? There are various processes. So types of DNA repair is mismatch repair, which fixes wrong base pairing. Then there is base repair, which removes damaged bases. Then there is nucleotide repair, which fixes larger distortion like UV damage. Okay. Then there is double strand break repair, which fixes broken DNA strand. Importance of DNA replication. What is the importance of DNA replication? Ensures accurate transmission of genetic information, prevents mutation and genetic disease, maintains genome stability, essential for growth, development, and survival. So, guys, basically, there are various importance of DNA replication and repair. As I said that DNA contains nitrogen bases. Also, nitrogen bases has its own importance. Nitrogen bases are crucial. Why? Also, why is DNA replication and repair crucial? Because nitrogen bases are crucial because they are core of genetic information. It stores genetic information. The order sequence of nitrogen bases in DNA determines the instruction for making proteins. Each set of 3 bases called codon codes for specific amino acid. Example, wait a second, guys. Example, ATG, start codon. Then also it helps in building proteins via RNA. DNA is transcribed into RNA, which is translated into protein. The nitrogen base sequence in DNA directly determines the RNA sequence. The resulting amino acid chain without correct base sequence protein wouldn't function properly. So yes, guys, importance of DNA replication repair: ensures accurate transmission of genetic information, prevents mutation and genetic disease, maintains genome stability, is essential for growth, development, and survival. So as we got to know, there's various importance of DNA replication and repair. We got to know different types of DNA. We got to know how what is the structure of DNA and what does the structure of DNA contains, what is it made up of. So these things are really important to know about DNA. These are the basics of DNA and the basics of DNA replication repair. If anybody understands this, they can clear the concept and move further to study DNA more profoundly. But first, in order to understand the more complex thing, we need to understand the basics of DNA, the basics of DNA replication, the basics of DNA repair, the nitrogen bases, what are its importance. Once we understand this, then we can profoundly understand the more complex process of DNA replication, DNA repair, and what and how else the DNA is being beneficial for all of us. So thank you guys so much for watching my video. Please like and subscribe my channel. Thank you very much.